This is Lab 16 associated with Lecture 16, and this is on PCA regression, or Principal Components Regression, PCR. So for this lecture, we're going to work with some new data, and this is from TCGA, which is the Cancer Genome Atlas, and I've curated a small subset of the TCGA data, and this is gene expression data. So it's a relatively small curated subset. It's going to have about 99 observations and 190, 200 or so variables, and the data is the collection from about 100 of these samples of gene expressions for these people. So here we have, say, gene 1, and this is the expression of gene 1, the expression of gene 2, expression of gene 3, 4, 5, etc. So gene expression is basically how active is that gene. So a little lesson in biology is that not all parts of the genome are used as much as other parts. And so some parts are used a lot, some parts are used a little, and so you can measure the amount that these various parts are used by measuring gene expression. And so this is called high throughput gene expression, and it's a very common thing to do in biology. And has been for the past 20 or so years. So this data, though, so each row you can think of as a person, and we have some number of genes here. We're just going to look at 200, and in a real person, you'd have 25, 50,000 genes. But one of the things about this data is that we have many more variables, those genes, than samples. It's very hard to collect people. It's very expensive, so we may only have, say, 100 people, but it's very relatively easy to measure nowadays 10, 20,000 genes at a time to do these gene sequencing. The technology over the past 70 years or so has been really refined. So this is a common problem that we have in this high, kind of what is called high dimensional problem, where the dimension, as the P, is really high compared to the N, which is low. Now this is not too bad of a case, but you will have data sets where you have 30 people that you're able to sequence the genome, as it were, transcriptome on, and measure 20, 30,000 genes easily for these people. So you have this incredibly high dimensional problem. So we're going to curate this data set a little bit, just because there's some columns with some zeros. So we're going to call, first we'll just convert it to a matrix. I'm just going to figure out which columns have all zeros by just seeing which ones of them the standard deviation is 0, and then I'm just going to pull them out. So our curated data set is actually still going to have 100 people in it, or 99 people, but we're going to throw out a couple of these genes, about 4 or so of these genes that don't have any variation, because that's just going to cause some grief for the demonstration. And then we need a Y and an X, and I'm just going to say we're going to predict the 5th gene for whatever reason. It's not super motivated here, but you could imagine trying to predict the 5th gene from the other genes. And maybe in a more realistic example, what we'd be predicting is well, you could do a classification setting where you'd be classifying cancerous or not cancerous based off the gene expression of the sample, or you'd be predicting blood protein level off of the gene expression data or something like that. And so we're kind of gonna make up an X and a Y, right? So the Y is just our response. So there are 99 of them, and some values, and the X's, is the other stuff, is our subset of gene expression matrix. So what we're first going to do is we're going to rescale this. So we talked a little bit about this for PCA or PCA regression, that oftentimes, not always, you want to center and scale your data. You can do that with the function scale. First thing I do here, though, is I grab the column-wise standard deviation and mean. So we're going to need that because when we do PCA regression, right, if we do it not just using a software package, we want to write it ourselves, which we will. It's very easy. We need to know what transformations we did to the original data here. So I'm just going to rescale this data. I'm going to save what the original standard deviation means were, so I could basically undo the scaling if I needed to undo it. So we do this, we're just scaling it this way. And I have no missing values, so everything's good. And I could plot like a histogram of, I don't know, x1, or not, it's not going to let me. Strange. Well, it's not going to let me plot that. That's strange. In any case, so the typical thing to do is we want to predict y, one of our gene expressions, from the other ones. So use regression, right? And as we said, this is kind of a high-dimensional problem, which is very common in real. I mean, this is real data. It's very common. And what do we know is going to happen when we do this, right? We're going to run into a situation where we have a higher p, a higher number of variables than we do of samples. And so your regression estimate, your beta hat's actually ill-defined. You have a non-invertible x transpose x. So we can run this, and we'll get, our intercept, and we'll get betas for all of our genes. But if we scroll down here far enough, we start to get NAs, right? And we've seen this before, and this is because beta hat is just ill-defined. So if we say X design, let's actually name our model here L mod, right? Okay, and so we can actually grab that design matrix or the model matrix from our linear model, and we could say kappa of transpose design, right? Something like that, and we get this huge number, right? Because this thing is basically not invertible. It's interesting. Kappa is actually going to read the help page on kappa. It must be estimating well, yeah, numerically it's not infinite, but it probably actually is. Not really sure why it's giving me an error there. That's disturbing. Okay, so, but basically it's not. It's very, 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 very poorly conditioned. So we can run PCR, which by using the package PLS, which actually stands for partial least squares, which is another method we might get some time to talk about. And there's a function in there called PCR for principal components regression. And of course, this is going to give me an error, isn't it? That's great. Let's see. Okay, restarting that seems to have fixed it. Anyway, so here we have this function PCR partial, which does just principal components regression, or principal component analysis regression, whatever we're going to call it. So PCR, and you treat it just like a linear model. You pass it in your formula. There's some other arguments we can pass it in. Let's see what's interesting. Scale, whether or not we're going to scale and center it, which I think default to true. Yep. Yep. And should be a n-comp, number of components to include the model when doing, say, PCR. So that's going to be what we're calling Q in the lecture. And it gets a bunch of other help stuff. So let's just scroll past this. So it can fit the model, say PCR mod. Calling PCR, we're going to predict y from x, and let's use 10 components. So that's kind of an arbitrary number to choose. Maybe we can choose a better number, SVD x. We can get the variances if we run a PCA on this. The variances are just those diagonal elements squared. And so we can plot those variances. We see something like this. So how many components do we need? It looks like there's a need maybe around there. Plot the cumulative sum of the variances. So one way that people often choose how many components to retain, because it's a bit of a tuning parameter you can choose, you actually kind of cross-validate it, is we want to retain a certain percentage of the variance, and so if I plot the cumulative percentage of the variance, I can see how many components do I need to retain to get, say, more than 80% of the variance in my data. And this is telling me 1. That seems false. Let's say which is our max. So 29. It says about 29 components will be about 80% of the variance. So you could choose that. So instead of choosing 10, maybe a more data-informed way would be to choose 29. Sure. And we can get a summary of our model. So that fit real quick because all it's basically doing is an SVD, as shown in the lecture. And it gives us some summary of if I include one component, the percentage of variance explained in X and Y, and in the training data. And so we see, yep, 80% of the X is explained if we include 29 components, which is as we claimed. So we can predict using PCR by calling predict. We tell it how many components, so it actually can predict anywhere between. If you don't specify this, it'll give you predictions for all the way from 1 to 29. It gives me my predicted Y's with this model, right? Why is that interesting? Because, right, if you remember, if you go back up to our original linear model, you couldn't even fit the thing, right? It gave you null beta hats. So we've actually just fit a model and it gives us predictions, right? You could do this with ridge regression, but here we're doing with PCR. So this is that was using the package, but we can also look at coding it by hand. So to do that, I would pull off, say, 29 components from my V matrix, right? I get V, XV is my Z, right? Maybe I should call this thing Z, right? Let's say, all right, so it has 29 columns, and it will have, whatever, 99 rows. Yep. And so one way to do this by hand, all PCR regression is doing is now I've calculated the Z, and basically got my principal components, my Z matrix. And as I claimed, all it's doing is I hand fit a model here. I just throw it in linear regression, and I predict Y from, instead of the X's, which I had before here, I'm now predicting from Z in my principal components. And I'll get this model where it's predicting from my 29 principal components. Right, so here are those betas. I can also predict from that handmade model, and my claim is that this gives the predictions from PCR regression are the same thing as the predictions from my hand-fit model of literally just running, calculating the principal component Z, and regressing Y onto those Z's. That is exactly what PCR is doing. So that PCR function seems like it's doing something fancy, but this is literally probably all it's doing behind the scenes. And lo and behold, they are exactly the same. They fall exactly on the 45-degree line here. These are exactly the same predictions. We can get our PCR beta out of this. If we're smart. Whoops, this should be 29, shouldn't it? Yep. And so this is our beta, what we're calling in lecture, beta hat, whereas the coefficients from this hand-fit model is what we were calling gamma. And so these are our betas from PCR. You could alternatively hand calculate these things, right? It's just X times beta, right? So beta PCR is basically the coefficient where if I multiply it by X and I add on my mean here, that should give me the exact same predictions. That's another way of calculating by hand, right? So this beta PCR, I guess the other way here would be, we had what was called gamma, but that's just X by Z by the coefficients of my hand-fit model. And assuming these are not going to be the right size, let's just make this a 29 by 1 array. By 29. What size do we need it? Don't do this to me. X by V. There we go. That was not X by Z. That's probably why it's giving me grief. Should give me the same thing. well, never do anything live, right? So the beta PCR, because we don't want to include the intercept here. We want to exclude the intercept. Exclude the intercept. That's why they have -1 here, and then it's add back in the mean. Doesn't really matter. It's just convenience for coding. And again, so that's another way of doing it. So this is the coefficients from the tangent model, what we're calling gamma, and as claimed, this gives us the same thing. So we can predict on new data. If I have some new data, and I can just generate, say, some normal data. So if I just generate, here's some fake data. I've just generated a sample observation, right? You could just call predict PCR mod, and you pass it in the new data. And we wanted to pass in 29 of these things. And on my new thing, it predicts 9.6, blah, 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 right? Or I could do this by hand if I had calculated my PCR beta and I have my X. I can do this by hand, and here is the moment of truth. Is it going to give me the same thing? Yeah, it does. So, so that's one way we can. That's all that's happening behind the scene. Is literally what's happening here. So that's the quick summary. Real quick summary of PCR regression, but it's literally. You should just keep in mind is because we can't fit this linear regression model, all we're doing is we're calculating the principal components, and we are just fitting the regression model instead of y on x here. We calculate the principal component z, and we fit the model of y on z. And that's basically what's happening. And it's a really, it seems simple. It's a really powerful technique, and we talked about in lecture how it's kind of related to ridge regression and on some kind of spectrum with ridge regression because it's kind of solving the same problem in a different way. Okay, we'll stop there.